Hello students, I am Priya Lagwankar. Today we are going to deal with one more very interesting topic, and that is cell and cell organelles. As here you can see on the screen, see there is a three-dimensional picture of a cell. All organisms, they are made up of cells, and of course the cells, they arise from pre-existing cells. All these are known facts to us. But were they known since ancient times to human beings, or when they became known to us? Let us try to explore this history. In the ancient times, cells were of course not known to humans. It was for the first time the scientist named Robert Hooke. He tried to discover the cell. How he did that? That is very interesting. Robert Hooke had made a very simple microscope. See, this scientist, he was there in 17th century. He had made his microscope. You can see it in this picture. And he had used an oil lamp to illuminate the object. Of course, because in the 17th century, electricity was not there. So, Robert Hooke had made this microscope, and his curiosity made him observe the cork section under this microscope. So what is a cork? See, cork is a soft wood of which the leads are made. See, here you can see the stoppers of bottle or the leads of bottle, they are made of this soft wood called as cork. So what Robert did, he made a thin slice of this cork and he observed it under this microscope. So what he found? He found such honeycomb-like structures in the cork under microscope. These structures, they were empty, and that is why he labeled them as cells. Actual meaning of the word cell, that is empty places. So he found such empty chambers inside the cork, and that is why he coined the term cell. But actually what he saw were the dead cells from that plant. And as those cells, they were dead, there was nothing inside the cells. But is it so in case of all cells? Obviously not. But this was not known, and gradually with the development of microscope, different things about the cells, they were revealed. See students, here there is another scientist named Antony von Leeuwenhoek from the 17th century only. Actually, he was not a scientist by profession, he was a businessman. But due to his curiosity, he had made a very simple kind of microscope. You can see the microscope in the picture. Here there was a lens, and here there was a place to keep the object. And how he used to observe the things, that is seen in the photograph. So what he did, Leeuwenhoek once observed a drop of pond water under microscope, and he was surprised to see some living creatures inside the water. Till then, students, it was not known to human beings that such microscopic organisms are also existing around them. Means whatever world was seen by our eyes, only that much world was known to man. But for the first time, Leeuwenhoek observed that drop of water and he found such microscopic organisms in it. Some of them, they were unicellular, and some of them, they were multicellular. But existence of such microscopic things, microscopic living things, was for the first time, it was known to man by this discovery. And this discovery further triggered the study of a cell. Leeuwenhoek was the first person to observe the living cells under microscope. See, before that, as I told you, only dead cells were observed by Robert Hooke. But for the first time, living cells were observed by Leeuwenhoek, and of course gradually that led to the further development in the research of cell. Now students, all this happened in the 17th century, but directly we are coming to the 19th century. In the middle period, nothing much happened in the field of cell research. But in the 19th century, these two scientists, Matthias Leiden from botany and Theodor Schwann from zoology, they work independently in the field of cell research. They tried to find out what living organisms are made up of, and they independently concluded that all living things are made up of cells for the first time. And not only that, they also concluded that any living cell is capable of doing basic fundamental things. What does that mean? That means any living cell is capable of surviving independently. For example, amoeba, it is a unicellular organism. But still that single cell is capable of doing basic life processes and it can exist on its own. So that is why cell is called as the fundamental unit of life. So these things, they were stated by Leiden and Schwann. There is one more contributor in the field of cell theory, and that is Rudolf Virchow. So Rudolf Virchow was a scientist who told for the first time that the cells, they come only from pre-existing cells. That means cells cannot be generated spontaneously anywhere. Of course, they come from pre-existing cells by cell division. He said omnis cellula a cellula. That means cells can come from pre-existing cells only. So that was also included as a point in the cell theory. So students, so far we have seen that the discovery of dead cells was made, then the discovery of living cells, then it was stated that the organisms, let it be unicellular or multicellular, they all are made up of cells. Cells come from pre-existing cells. But the research work so far, it was confined to the cell only. Why? Because powerful microscopes were not available till that time. But as the microscopy developed, the scientist further studied what is inside the cell now. So for the first time, Robert Brown was a scientist who observed that there is a nucleus inside the cell. Now in the picture in front of you, you can easily see the nucleus. Not only the nucleus, but the nucleolus inside the nucleus also is seen by you. But this was first time seen by scientist Robert Brown. Then gradually the other organelles in the cell, they were also discovered. But it was only after 1920. Most of the cell organelles, they were revealed, their presence was detected. Why after 1920? Because in 1920, discovery of electron microscope, it took place. Electron microscope can magnify an object 1 lakh times. And that is why the structures inside the cell, which could not be seen even under compound microscope, they were now revealed. Here you can see, students, this is the cell wall and cell membrane, and inside that a fluid, you can say a thick fluid which is filled, that is called as cytoplasm. And there are different structures floating in that cytoplasm. And those structures, they are called as cell organelles. So which different cell organelles are present in the cell? Let's see them. So we have already seen the nucleus in the cell. Nucleus is most distinctly seen cell organelles, and of course it is the controlling center of the cell. Like our brain controls the functioning of our body, the nucleus, it controls the functioning of the cell. Then there are organelles like ribosomes, rough endoplasmic reticulum, smooth endoplasmic reticulum and Golgi apparatus. See, all these organelles, all these 4 organelles, they are related to synthesis of proteins and their transport. Then students, this chloroplast is an organelle, of course, which is seen only in case of plant cells. This chloroplast absorbs light energy and helps the plant to carry out the process of photosynthesis. Now, next is mitochondria, singular mitochondrion. So, mitochondrion is called as the powerhouse of the cell. Why? Because formation of energy which is required for the cell or for the body, that is made in mitochondria. Here, the cell membrane, it is shown, its microscopic structure has been shown here. The cell membrane, of course, it binds the cytoplasm and it protects the inner part of the cell. Then here there are lysosomes and vacuoles. See, lysosomes are responsible for digestion within the cell and they also protect the cell from some microbes if they enter inside the cell. Then this vacuole is a membrane bound sac-like structure in which storage is done in the cell. Then there is one more organelle seen, centriole, that is helping in the cell division. In this way, students, different organelles are seen inside the cell. But the question is, are all organelles present inside all the cells? The answer is no. Depending on the presence or absence of such organelles, we can classify the cells. That is, prokaryotic cells and eukaryotic cells. So, prokaryotic, that means primitive cells. These cells do not show presence of such different cell organelles. In that cell, only you can say a cell membrane, cell wall and a capsule around the cell wall is present. Then the material like DNA and RNA, it is not present inside the nucleus, it is directly present in the cytoplasm. So such simple structure is seen in a prokaryotic cell. Whereas in case of a eukaryotic cell, students, there are different cell organelles which we have seen. Such cell organelles are seen in the cytoplasm of a eukaryotic cell. Eukaryotic cell is of course larger in size than a prokaryotic cell. Then how come the cells are of these two types and how they were made? Actually, you know, even if I am telling you that the cells are coming from pre-existing cells, students, millions of years back, when the first cell was formed on earth, it was a very simple type of cell and it was formed accidentally from non-living things. Even though it is not possible again, but that cell was formed at that time and that cell had a very simple structure. It was just a piece of nucleic acid covered by a cell membrane. So that was a very primitive type of cell, even simpler than the prokaryotic cell which you are seeing on the screen. So gradually that modified and gave rise to the prokaryotic cell. Then from the prokaryotic cell, gradually eukaryotic cells were formed. How? That we are going to see. Organisms like bacteria, they are having prokaryotic cells. Their cells are of prokaryotic type. So in ancient times, long back, the bacterial cells which were present on earth, what happened? Some bigger bacteria, as you can see in the diagram, some bigger bacteria, they engulfed some smaller bacteria. Okay, they engulfed some smaller bacteria. They are called as aerobic bacteria because they required oxygen for their survival. So they are called as aerobic bacteria. And what happened? Those smaller bacteria, they lived inside those bigger bacteria. It continued for a long time. They started living together symbiotically. That means they helped each other and they lived happily. So as it continued, the smaller bacteria, they became a part of the body of the bigger bacteria. And gradually, as you can see in the picture, they were converted into mitochondria. Then similarly, by some bigger bacteria, smaller bacteria called as cyanobacteria, which contained chlorophyll in it, they were engulfed by these bigger bacteria, and gradually that led to the formation of plant cell, whereas the bacteria with just mitochondria in them, they gradually evolved in animal cells. So that is how two types of eukaryotic cells, animal cells and plant cells, they were formed from the prokaryotic cells. Now let us see, students, who all are coming under prokaryotes and who are included in the group eukaryotes. So as I already told you, all the bacteria, their cells are of primitive type. Of course, all bacteria, they are unicellular and their cells are having primitive structure. So different types of bacteria, they all are included in the group prokaryotes. Whereas the eukaryotic group, the organisms having eukaryotic cells, they can be unicellular or multicellular. The unicellular eukaryotes, they are included in the protozoa group. Here you can see they are included in the protozoa group. Whereas multicellular eukaryotes, we classify them as plants and animals. So in this way, students, the cells present in our body also, they are eukaryotic cells. So we are eukaryotes, we are multicellular eukaryotes. Let us see the similarities and differences between a plant cell and animal cell. You can mark which structures are present in both of them. So structures like nucleus, Golgi complex or mitochondria, they are seen both in case of plant cell and animal cell. But which structures are absent in animal cell? Of course, the chloroplast. See, the chloroplast here, which is present in case of plant cell, of course it is absent in animal cell. And that is why animals are not capable of making their own food by photosynthesis. There is one more very important structure present in plant cell and absent in animal cell, and that structure is cell wall. See, here in case of both cells, the cytoplasm, it is bounded by a thin membrane that is called a cell membrane. You can see here there is cell membrane in the plant cell, and cell membrane is present in the animal cell also. But outside the cell membrane, here in case of plant cell, there is one more binding structure, and that is called a cell wall, the one which is shown by a thick green line. See, this is the outermost covering in case of plant cell, and that is called as cell wall. So cell wall is absent in case of animal cell, and due to presence of cell wall, the plant cell gets rigidity. So the plant cell is more rigid and having a definite shape due to presence of cell wall. Students, have you pointed out one more difference in the two diagrams? Here in case of plant cells, there is a large vacuole. Such large vacuole, it is not seen in case of animal cell. In the animal cells, vacuoles, if present, they are small in size and they are many in number. Whereas plant cell always has a single vacuole and it is very large in size. So these are the main differences in case of plant cell and animal cell. You can observe the cells and find out if any other differences are there in plant cell and animal cell. Now, students, so far we have seen the journey of discovery of cell, then the types of cells, that is prokaryotes and eukaryotes, and in eukaryotes again plant cell and animal cell. So now let us see the comparative sizes of different cells. What is the size of each cell? So here students, you can see the scale. First let us try to understand about the scale. See, as we go from right to left, you can see here millimeters, then what here it is written is micron, and then nanometers. So what is a micron, students? Students, you can take a scale and observe on it that 1 centimeter is divided into 10 equal parts, and each part we call it as a millimeter. Okay, so how small 1 millimeter is? Now that 1 millimeter is further divided into 1000 equal parts. See, because these structures inside the cell, they are very small and they cannot be measured, of course, in millimeters also. So we require smaller units. So that is why 1 millimeter, it is divided into 1000 equal parts, and each part is called as 1 micron or micrometer. So here you can see the micrometers on the scale. That is also not enough. Some structures, they are even smaller than that, which cannot be measured even in micrometers. So further 1 micrometer, it is also divided into 1000 equal parts, and each part, it is called as a nanometer. So these micrometer and nanometer are the units to measure the cells and cell organelles. So let us see, students, the comparative sizes of different cells. Let's begin from the extreme right. So what we are able to see here, see, there is ostrich egg, there is egg of chicken, there is frog egg, and even human egg. So you will say, why these eggs are shown over here? See, when these eggs are in unfertilized stage, these eggs are also examples of a single cell. That means ostrich egg is the largest single cell. Ostrich egg can be considered as the example of largest single cell. So what is the size of that ostrich egg? You can see here, its diameter is around 100 millimeter. Then gradually coming towards the left, you can see that the diameters are lesser, and finally general animal cell or general plant cell. What is their size? Of course, we have to observe them under the microscope. That means their size is smaller than 100 micron, because objects greater in size than 100 micron, they are seen by our naked eyes, but we cannot see the cells by our naked eyes. So of course their size is smaller than 100 microns. So any general plant cell or animal cell, its size lies between 10 microns to 100 microns. Further going to the left side, here you can see the mitochondria, and here you can see the bacteria. Bacteria, that means the prokaryotic cell. So as I already told you, the prokaryotic cell is of course smaller in size than the eukaryotic cell. So the size of cell organelles like mitochondria or the size of a prokaryotic cell is around 1 micron. And the contents inside the cell organelles or any molecules, they are even smaller than 1 micron. And whatever structures, they are smaller than 100 nanometer, they have to be seen with the help of electron microscope. With the advancement in the microscopy, more and more structures inside the cell, they are revealed. More and more research work regarding the cell, that is going on. So students, today we have seen how the cell was discovered and how it is classified and what are the structures present inside the cell. I hope you have found this interesting. Today, students, we are going to stop here. Next time, I will come up with some new interesting topic. Till then, goodbye students.